Hello, good morning everyone. I am the instructor of this exciting journey of protein structure and engineering, which is chapter 2 of unit 5 of your instruction source. So, my name is Bishwajit Kundu and I am a professor at the Kusuma School of Biological Sciences at IIT Delhi, and I am excited to deliver this series of lecture which I will be covering in about 5 separate lecture series. So, to begin with, about protein structure and engineering, let us look at what these proteins are. So, if you look around yourself or the surroundings, for example, all the plants, animals, bacteria and even the viruses, they contain about 15% of protein in their total mass. These proteins have various functions, and as has been rightly said, they are called the workhorses of the cells. So, for example, you can see they can function as enzymes, where you can see an example, a very classic example is Rubisco, which is an enzyme, very important enzyme of the plant that converts light energy into chemical energy. You have storage proteins, for example, ferritin, which stores iron, a necessary component of all the cells. You can have structural proteins, for example, the spider silk, or for that matter the wool, or the hair, and many other such structural proteins. You can have receptor proteins. The simple fact that you have been able to visualize certain things or differentiate between colors through your eye is because of the presence of this protein called the rhodopsin, which acts as receptor in determining or deciding your color. There can be motor proteins. So, when you are listening to this lecture, probably you are blinking your eyes, and when I am moving my hands and legs, or then also I am actually doing a lot of motor work, and all those are also done by motor proteins such as actin and myosin. You can have signaling proteins, for example, hormonal signaling like insulin, which actually helps in controlling your sugar content. You can have proteins for protection, which includes immunoglobulin, and immunoglobulin you might have heard about the presence of antibodies for the SARS coronavirus. So, whether you have the antibodies or not, that is being detected, and whether you have got infected with coronavirus also gets verified by the presence of this immunoglobulin. Then you can have transport proteins, for example, the hemoglobin. So, hemoglobin carries oxygen, so this is a transport protein, and then you can have various other things like gene regulatory proteins and some very special function proteins, which I will talk to you in the course. So, we proceed to understand what these proteins look like, and they actually look like this. So, it is almost like a chain or a string, and in those string you can have these small beads. So, these small beads as represented in this string are actually the amino acids. So, these are the amino acids, one amino acid connected to another amino acid, connected to the third and the fourth. So, here is a list of all the amino acids that are present naturally. There are 20 of them. Now, if you look much closely, what are these amino acids? The name itself suggests that an amino acid has something called an amino group and a carboxylic group or an acidic group. So, you have amino group and acidic group. So, that is called an amino acid connected to a central or C alpha carbon atom, and this C alpha carbon atom right here is connected to a different kind of side chains. So, while this backbone, that is an amino, C alpha and a carboxylate group with this hydrogen atom will remain continuously like a backbone, this R group, which is called the side chain, will keep changing, and depending on what this R group are, these are the different names of amino acids that are given. For example, if you look at this list of amino acids, so you can see that there is an amino group, there is a carboxylic COO group, and this central carbon atom with the hydrogen, which is exactly replica of this, and then if you replace this R with a hydrogen, then that becomes glycine. If you replace this R by a methyl group, then it becomes alanine. If you do so and go on, like for example, you have valine, you have leucine, you have isoleucine. So, you can see there are large number of these chains which vary in the chain length and the kind of branching that happens in these chains. So, you can have a methionine, you can have a phenylalanine, tryptophan, proline, etc. So, these are the different amino acids. Now, they have been categorized depending on their specific functions or the specific properties of these R groups or the side chains. So, for example, these green colored ones are representative of non-polar side chains. That means they do not react with water, whereas they do not interact with water and therefore, they are unable to form hydrogen bonds with water, a property which I will talk to you later on. If you look at some other side chains like these pink ones, so you can see this R group is replaced by CH2OH or CHOH, and so on. So, these are the polar side chains. That means there is a possibility of formation of hydrogen bonding with water. That means they are soluble or miscible in water. You have some other kind of amino acids which are electrically charged, having electrically charged side chains, which can be acidic and therefore, they are negatively charged, or basic, therefore, they are positively charged. For example, aspartic acid or aspartate and glutamic acid or glutamate, these are acidic amino acids. And then you can have basic amino acids which have, as you can see, an ammonium group attached at the end. For example, the lysine, the arginine, the histidine, and so on. So, what do you have? Any combination of these chains. So, for example, you can have here, for example, you can have a phenylalanine, that means this R chain is this, and then you can have a leucine, for example, and then that R chain is something like this, and then you have a serine, okay, which has an R chain like where is that? Serine here, and then you have a cysteine and so on. So, cysteine is here. So, you can have one after the other these amino acids, and these amino acids are connected through this nice, you can see this thread here, which is called a peptide bond in our case. We will look at what peptide bond are in the next slide. So, let us go back. As I said just now that the proteins are made up of long chain of amino acids, and I have just now told you how an amino acid look like. The R chains do vary, and depending on that you have these different types of amino acids. Now, let us say there are two amino acids coming next to each other. So, let us imagine a position here. So, this is one amino acid, and the next one, which is the next amino acid, or maybe any other amino acid at the center. Let us say this is phenylalanine and this is leucine. Now, what happens when these two amino acids come in close proximity? So, there is a carboxylate group or a acidic group, and there is an amino group, and therefore, a dehydration reaction simply, or a condensation reaction between these two with elimination of water molecule, as you can see here, will result in the let us say we have another group which is coming next, another amino acid coming next, and then similar kind of reaction between this carboxylate group and this amino group happens, and there is elimination of water. So, two H groups, H2 and O. So, water molecule goes out. Therefore, there will be a link that gets formed between the C double bond O NH and again C double bond O NH. So, you have CONH, CONH. This kind of group binding is, or a bond, is called a peptide bond. That is what happens. So, you have amino acid number 1, which is shown here. You have the R group which differs, and then you have C double bond OH. This OH reacts with H here and then results in the formation of what is called as a peptide bond. So, you can keep on adding as many amino acids as possible by formation of consecutive peptide bonds. So, 1 peptide bond, 2 peptide bond, 3 peptide bond, and so on, and then you can generate what is called as a polypeptide bonded structure, which resembles a bead on string. So, while this is very exciting, you will be surprised to understand, and you will be excited to understand how these peptide bonds and the corresponding R chains actually help this polypeptide to fold. But before that, I would like to tell you that when there are 3 amino acids connected by 2 peptide bonds, this is, however, although there are only 2 peptide bonds, 1 and 2, this is still called as tripeptide. That means 3 amino acids are connected by 2 peptide bonds. That is called a tripeptide. If there are 4 amino acids connected by 3 peptide bonds, that will be called a tetrapeptide, and so on. Now, let us move on to figure out how this protein, that is a polypeptide, assumes certain structures, because as you can understand, so if you have a long thread like this, you might have seen threads at your home. So, if you have a long thread like this, it is like a flippy, floppy, moving around noodle, and you can just leave it like this in the room, then it will form different kind of folded structures or wrapped around structures like this, which is kind of the same formation happens in the case of these polypeptide chains which are shown here. So, when it folds, however, in this fashion, that is, it takes certain shape, these shapes are not random. The unrandom or the non-random behavior of these shapes is what determines the function of a protein, and that is what the exciting story I will be going to tell you in the next few slides. So, let us move on to the next slide now. As I have been telling, look at this particular structure here. So, you have amino acid number 1 and amino acid number 2. These 2 amino acids are connected because of the dehydration reaction by the formation of a peptide bond. Now, here is a peptide bond that has been formed. So, you have CO right here, CO, and then NH, which is right here, NH. Now, we already know that oxygen is an electronegative atom. That means it tries to pull a pair of electrons towards itself, and nitrogen, after formation of these bonds, has a lone pair of electrons. Now, what happens when this oxygen, which is an electronegative atom, tries to pull this pair of electrons towards itself? This vacancy is being filled by the presence of these lone pair of electrons from nitrogen, as a result of which, in a quick transition, this oxygen assumes a partial negative charge, whereas this carbon and nitrogen, actually there is a formation of a partial double bond structure, a double bond character, which was otherwise a single bond. Now, in this situation, the nitrogen is now electropositive, so it has lack of electrons. So, what might happen in the very next moment is that this nitrogen will try to fulfill this electron vacancy by pulling this bond again back. As a result, there will be a quick tautomeric comparison, structural shift, resulting in the formation of a single and a double bond between this quickly, and that is what causes a transient, what we call as transient double bond character or partial double bond character between the C and the N. So, this is the COO of one amino acid, COO of one amino acid, and N of the second amino acid connected by a peptide bond, and this peptide bond has a partial double bond character. Now, whenever there is a single bond, you know that rotation around this single bond is possible. But when there is a double bond, then the rotations are restricted, they cannot move around anymore, because this, although moves, the other one will restrict its movements. So, this is what happens in the case of a polypeptide. As I said, the folding of the polypeptide or the noodle is not very random. It is actually monitored or governed by certain simple physical rules, and one of that is the formation of this partial double bond character. So, what happens is this partial double bond character results in the formation of this particular blue shaded portion as a flat. So, as I have been saying, you have this flippy floppy noodle like structure, but it folds if you just leave it around in the system, but then it does not fold just randomly. There are actually various physical and chemical forces that determines how it folds, and it folds into the exact similar structure 100 times you unfold and refold or unfold and refold this. So, that happens because of some physical and chemical forces. We will look at what are the determining factor that decides some of the folding properties of this polypeptide chain. So, the next slide, here I would like to tell you, look at this situation. So, you have amino acid number 1, amino acid number 2, amino acid number 1 has this carboxylate group, amino acid number 2 has this amino group, and there is formation of a peptide bond as we call, because of the dehydration reaction, and this peptide bond now is here. So, you have CO which is represented here and then NH which is represented right there. So, if you have a CO and NH, then what happens is this O is an electronegative atom, and as you know, an electronegative atom has the propensity to actually pull a pair of electron towards itself. So, it tries to pull this pair of electron which is forming this C double bond character towards itself, and in the process this actually gets a vacant position. Therefore, this pair of electron pair which is present on the nitrogen atom gets pulled to form a bond right here. Now, in the very next moment, this N, which is now a positively charged atom, will try to pull this pair of electron back to fulfill its own octet, and as a result of which this oxygen will again, this pair of electron will again be pulled back to form this double bond. So, what happens, there is a continuous shifting of this partial double bond character between the O and the N, which results in the formation of what we call as a partial double bond character between C and N. So, if you can see, if you just cut it across right here, then you have the C right there, C alpha, the C double bond O which is right here, CO, and then you have NH which is represented from here. But remember, this NH is coming from the next amino acid, but the amino acid number 1 which cut across between these partial double bond character, that is this sphere, is now connected to this sphere by a partial double bond character. Now, this is what creates the first restriction in terms of the mobility and randomness of the folding of this polypeptide chain, as I was talking to you. So, what it does is, as you know, if there is a single bond, then there is free rotation all around possible, but if it is a double bond, then the rotation of these molecules are not possible. So, what is happening essentially is that once this peptide bond is formed, this and this actually behaves like two planes. So, this is one plane, this is another plane. Now, this is connected together, which is shown by these light colored boxes or the flats. So, these are two planes. Now, rotation around these two planes are possible, but within this plane it is not possible because there are multiple bonds. So, that is the first set of restriction that is applied. We will proceed to look at what are the other restrictions in later duration. So, the other restrictions obviously will be the presence of this R group. So, sometimes this R group may be positively charged, sometimes it can be negatively charged, sometimes it can be polar, sometimes it can be non-polar, and so on. So, just imagine you have a positively charged R group here, let us say lysine, and then you have a negatively charged R group here or later down somewhere in the series. So, if that happens, and then there is a possibility of this entire process wrapping around like this. So, you have a positively charged amino acid R group hanging right here, and then there is a negatively charged R group hanging right here. Although this flat plane is not is rotatable against this flat plane, but then this flat plane again rotates against third flat plane, and so on. So, there is possibility of this positively charged group interacting with a negatively charged group down the lane somewhere, and that is how it decides how a polypeptide would gradually fold and take some spatial structure. But we will come back to the folding of those proteins in later course. For the time being, let us proceed to the next slide. Whatever be the constraints by which the protein forms finally a folded structure, it actually takes various kinds of 3D shapes. So, if you look at the possible 3D shapes, so you can have a cylindrical shape, a hexagonal shape, a torus shape, a cone, and so on and so forth. So, you can have various kinds of shapes, And to our surprise, nature has created these beautiful shapes, perfectly replicated these beautiful shapes in the form of the folded proteins. So, when this structure is being shown, you can see this is a beta barrel structure, where a polypeptide, which is this flippy noodle like structure or the beads on a string like structure, has actually taken certain shape to give rise to a beta barrel structure, which is more like a cylinder. If you look at the HIV, that is a virus, so HIV on its surface has various proteins, and the proteins are very nicely organized in the function in the structure like this. This is called HIV capsid protein, which almost looks like a tetragonal or dodecahedron structure. If you look at the very fresh and the one that we are now talking about, the coronavirus, you might have seen the structure or the figures of coronavirus, which is a sphere, and on top of those spheres there are some protruding things. So, those protruding things are called the spike proteins, and it is with these spike proteins that the coronavirus actually attaches to a host cell. So, this is a structure of a spike protein. As you can see, it looks more like a triangular structure or a tetrahedron structure, which is shown here. So, all these structures are fantastically represented in nature, which are originally there are these possibilities. So, as you can see here is entire list of various kinds of structures that is possible. These are just a subset, there are many more, and in every case the figures I thank those who have actually generated these figures, and I have given the references to each figure from wherever this has been taken. So, you can see there are various proteins which have taken various shapes. Some are hemoglobin, the carrier proteins, there are catalases, that are enzymes, there are structural proteins like collagen, and then there are purines, and there are cytochrome. So, all kinds of proteins are there. They are actually a polypeptide, that is this thin string, which has just folded and wrapped around and formed some 3-dimensional structure, and that structure is visible here for your reference. If we move on, how do we understand, how do we learn this structure? So, for our understanding, the protein structure has been divided into something called a primary structure, a secondary structure, a tertiary structure, and a quaternary structure. So, let us look at what these structural compositions or hierarchy is. So, if you look at the primary structure, which is shown right here, primary structure is nothing but the simple sequence of amino acid in a polypeptide chain. So, if you have, as I have been telling you, the polypeptide chain, which is going from one end to the other with bead on string kind of appearance, and the peptide and the amino acids are like glycine, proline, threonine, glycine, and so on. So, you have a primary structure, that is the sequence of amino acids. So, this is, for example, one protein which I will tell you in due course, and then you have another protein, for example, this insulin, which you can see you have glycine something and so on. So, these are the amino acid sequences. So, one amino acid sequence connected to another amino acid sequence by formation of some disulfide bonds in between. So, this is insulin, one of the proteins. This is another protein which is a part of a hemoglobin, and then what is shown here is that there is a glutamic acid right at the 6th position, which gets converted to valine, which is another amino acid. So, when a glutamic acid, that is an acidic amino acid, gets converted to a valine, that is valine replaces this glutamic acid, then the structure of this protein is completely changed. It becomes what is called as sickle cell hemoglobin. So, hemoglobin becomes sickle celled hemoglobin. We will look at it in due course once again. So, whatever it is, the primary structure is just the simple amino acid sequence, that is the beads. What are the beads? A, B, A, D, C, A, whatever, that is the sequence of the amino acids. And remember, there are 20 of these amino acids, so they can come in any different combinations. So, 5 A's followed by D, A, Z, Y, X, whatever, So, that sequence itself is what is called as the primary structure of the protein. Now, let us move on to secondary structure of proteins. So, if you have a primary structure of the protein, which is again the beads. As I told you, these beads on the string can actually be wrapped around, let us say, a pen, and then what you see is actually a helical structure. So, if you take it out, it probably will still remain a helical structure. Let us see if it can. I do not know, but no, whatever, you know. So, we can assume that it is kind of a helical structure. So, it can form a helical structure, and that is one of the secondary structures, or it can form a different kind of structure where some beads are flat like this, you know, solid flat like this, and another set of beads actually fall across this line, and then another set of beads fall across that line. So, you know, you can have this kind of a structure which is called a beta pleated sheet. So, these are sheets of amino acids connected by the single, you know, the peptide bond. So, you can have a twist like this, so that is alpha helix. You can have a flat structure like this, which is called a beta sheet, and so on and so forth. So, there are many such secondary structures. So, the primary structure, you remember, is just what are the amino acid sequences. Secondary structure is how these amino acid sequence actually form a twisted or a turned structure, alpha helix, beta pleated sheet. So, how this sequence is twisted or folded constitute a secondary structure. So, for example, an alpha helix, which is shown here, this is an alpha helix, is a coil of amino acid and are held in place by hydrogen bonds. So, there are hydrogen bonds that are formed between one portion of the amino acid or the polypeptide with another portion of the polypeptide, which we will probably look at in much detail later on. A classical example of such kind of hydrogen bonded alpha helical structure is, as I just now told you, wool. So, this is a structural protein, wool, which is obtained from sheep. So, this is the structural protein, and these are stretchy protein that is mostly alpha helical in nature. Now, as I said, there can be beta pleated sheet, which is shown right here. So, a beta pleated sheet consists of an extended chain of amino acid, which I just now showed, and one classical example of beta pleated sheet is the silk. See, it is obtained from the silkworm. This is also a protein. So, this is obtained, this actually is formed by, you know, formation of this kind of structure, where the amino acid sequence are laid against each other in concentric flat, you know, ribbon like sequences. And then you can have a whole lot of these sequences combined in some other proteins. For example, here is one protein which is shown. So, you can see there are alpha helices shown in green, and there are beta sheets which are shown in red. But remember, this is just one particular amino acid as is shown here. So, you have one particular the polypeptide sequence which can fold into an alpha helix or a beta sheet, and then again a alpha helix and then a beta sheet, and a combination of all of those can result in the formation of a complete functional protein. In this particular two examples, wool and silk, these are primarily alpha helical in the case of wool and primarily beta sheet in the case of silk. But then remember, these are not the only type of structures that are possible. It can form a combination of these structures, which we will look at in later slides. So, now we have understood primary structure, which is the sequence of the amino acid, the secondary structure, that is how the sequences first fold and form some kind of a twisted configuration, whether alpha helix or beta sheet. And now we will come to the tertiary structure, which I just now said is a combination of alpha helix and beta sheets that constitute the entire polypeptide chain. So, let us think, you know, that this is the entire polypeptide chain, and as you can see in this structure, this polypeptide chain goes in twists and tumbles and then forms some alpha helix somewhere, that is one of the secondary structural components, and then some beta sheet somewhere else, that is another secondary structural component, then again an alpha helix, then again an alpha helix, some beta sheet, helix. So, it starts from one end, continuously goes all the way to the other end by formation of different kinds of alpha helix, beta sheet, turns and twists. So, that is the secondary structure, the tertiary structure. So, what is tertiary structure? Tertiary structure is how the twisted chain is folded into a compact structure. Tertiary structure is held in place again by hydrogen bonds and many forces like the hydrophobic forces, disulfide bonds, hydrogen bonds, ionic bonds or the salt bridges and so on. So, these are the different kinds of forces which are called the non-covalent forces, a subject matter of discussion in my next class, and I will be talking to you in greater detail about these kinds of forces which hold the different the polypeptide chains in place. In these tertiary structures, very often what happens is the non-polar groups. So, when I say non-polar groups, that means there is no electronegativity, no electropositivity separately. All the groups are these groups are incapable of forming hydrogen bonds with water, and therefore, they can also be called as hydrophobic groups. When I say hydrophobic, that means they have phobia, they have fear against water, and therefore, they do not want to interact with water. So, non-polar groups, you remember each one of these polypeptide is made up of amino acid, and the amino acids have the R groups, and the R groups can be, so R groups are hanging somewhere here, and those R groups can be hydrophobic, that means they do not want to interact with water. So, if you imagine this polypeptide which has folded, twisted and formed something, and then you can think of a ball, you know. So, a thread has been just wrapped around and formed in the form of a ball, represented in the form of a ball. Then there will be some portions of this polypeptide which are towards the core inside of the ball, and some other portions which are exposed to the outside of the ball. So, those which are actually hidden in the core of the ball in a folded polypeptide 3-dimensional shape, those portions are naturally hydrophobic because the entire rest of the environment is hydrophilic. It is full of water in a cell or any other environment, but inside it is actually compact and no water can go inside. So, that is where the hydrophobic amino acids will be. So, that is tertiary structure. Now, we proceed on to another level of structural composition, which is called the quaternary structure. What is quaternary structure? So far we have been talking about the structure of a single polypeptide. A single polypeptide, when you look at just the amino acid composition, that is called a primary structure. When it forms some minuscule structures like an alpha helix, that is a secondary structure. When a combination of alpha helix, beta sheet, turns and twists come together and form some kind of a jumbled structure, but it is still a single polypeptide chain, it is called a tertiary structure. And now we are coming to quaternary structure. What it means is many proteins cannot function individually as a single polypeptide chain. So, what happens is you have a single polypeptide chain which folds, twists and turns and forms a structure. Now, this structure combines with another polypeptide chain which has independently formed another structure, and these two structures now come and combine in a given 3-dimensional orientation, and that orientation now becomes functional. I will give you an example and also tell you that is what is called as a quaternary structure. So, here is an example. As you can see, each color here represents one polypeptide chain. So, yellow color shows one polypeptide chain, the red color shows another one, the third one and the fourth one, and in each polypeptide chain you can see they have actually essentially folded into an alpha helical configuration. So, this polypeptide chain has folded into alpha helical configuration. So, this is the tertiary structure of this yellow polypeptide chain. This is the tertiary structure of the magenta, blue polypeptide chain. This is the red one and so on. So, there are 4 polypeptide chains, but now each one of these polypeptide chains have come together to form a quaternary structure, and our very well known hemoglobin is such an example. So, what are quaternary structures? When many proteins actually assembles or assemblies of multiple polypeptide chains, the quaternary structure is formed, and they refer to number and arrangement of the protein subunits with respect to one another. For example, as I told you, hemoglobin is a classical example. So, there are 4 polypeptide chains in hemoglobin. They come together and hold a particular position called the heme group, particular functional group called the heme group, and that heme group actually is responsible for holding of iron, iron, and that iron is responsible for carrying oxygen. So, hemoglobin is one such, DNA polymerase another enzyme is one such example, and ion channels are again examples of quaternary structures. So, with that, now that we know what are the different kinds of structures that a protein can form, we will move on to the next one and talk to you about some very interesting facts about proteins. So, two very interesting facts about proteins is that, as I told you, just imagine a kid playing with a thread and then he somehow jumbles up. You might have flown kites in your childhood, and then sometimes the thread of the kite gets jumbled up. If you try to unjumble, most often what happens is that will form various kinds of twists and turns, and it will form knots, and then it will be very difficult for you to actually open the knots, right. A polypeptide is exactly like this. So, you have a thread which is made up of some beads, and you just leave it in the environment, it will just fold in some order, and then this folded structure can actually, if you pull like this, it might simply end up into formation of some kind of knots. But interesting thing about proteins is that such kind of knots are very rare. I will tell you some other examples, some other interesting facts. So, as I told you, you can just have a look at this particular figure. So, you have a polypeptide which starts from one amino group, and this carboxylate group is attached to the amino group of the proline. The carboxylate group of proline is attached to that of amino group of threonine, and so on, so that this polypeptide is formed. And as you can see, there is an amino group at this end, and the end amino acid will have a carboxylate group at the other end. Therefore, they are called the end terminal, that is amino terminal end, or a carboxyl terminal, or the C terminal end of a polypeptide. So, you have an N and C terminal end of the polypeptide. Now, you also know that this NH group, NH3 group, is capable of reacting with the COOH group of the next one. So, you have the COOH group of glycine interacting with NH group of proline, COO group of proline interacting with NH group of threonine, COO group of threonine interacting with NH group of glycine, and so on. So, if you proceed in that fashion, you have the COO group here. Now, imagine a situation when here is the N group, NH3 group, and here is the COO group of the last amino acid. Now, in the process of jumbling up or folding, it is quite possible that the NH3 group of the first amino acid comes in close proximity with the COOH group of the last amino acid in a process something like this. So, here the NH2 group has come in close proximity with COOH group. Now, what is the possibility here? Because this has a tendency to interact with COOH group, it would interact, would not it? And if it interacts, then what happens essentially is that this polypeptide becomes a circular polypeptide, because this end is now reacting or interacting with this end. But in nature, circular polypeptides are, again, there are hundreds of examples, but there are not thousands. So, we have about 30,000 different proteins available in any cell at a given point of time in human cell, but then there are these circular polypeptides are rare. Here is a list of some of the discovered circular polypeptides. Until 1970s, they were not known, and these were rare events. They have different functions. Their sources are also mentioned here. So, you can go ahead about looking at these. This is obtained from Trends in Biochemical Sciences. A review has appeared in the year 2002. You can have a look and wonder about how these things are formed in nature. As I told you, the next interesting fact about protein is the knotted protein. So, here is an example. So, you have the N terminus of one protein, the C terminus of the same polypeptide, and the N terminus somehow goes through the interface of the loops of the same polypeptide and forms a knot. In nature, these kinds of knots are rare. They are there, but then are rare. But the question is why it should be rare. most of the polypeptides that you come across, most of the proteins that you come across, if you just have a jumbled up structure, you hold one end, hold the other end, pull it apart, it will never form a knot, except these few examples which are shown here. So, these are very, very interesting things. So, one of these, for example, is human ubiquitin C-terminal hydrolase isoform 1, also called UCHL1, is 52 knotted and accounts for up to 5% of soluble proteins in neurons. There are 52 knots in this protein. And if you have an unknotted version of the same protein, that is UCHL1, then the unknotted one, however, has been implicated in Parkinson's disease. So, let me now bring back to you about some of the problems that might happen in the process of the polypeptide folding. So, ideally a polypeptide gets folded into a specific three-dimensional structure that is dictated by the presence of these amino acids. So, each amino acid has a specific charged group, non-charged group, polar group, non-polar group, and so on, and those dictates how this polypeptide will fold, whether it will fold into an alpha helix, whether that alpha helix will react to a neighboring alpha helix, and so on. So, while that is dictated, and therefore, a given polypeptide will fold into a single specific structure every time that you do so, or that it is allowed to do so. However, problems do appear inside the cell. What are the problems? Problems are of various folds. For example, if somehow, let us say this polypeptide is supposed to form an alpha helix, but because of some constraint which could be shock, temperature, acid, or any other thing, this polypeptide is incapable of forming this particular structure at that particular moment of time when it is synthesized. Now, what happens is, instead of forming this kind of a structure, it forms a different kind of structure, and therefore, that kind of a different kind of structure is called a misfolded protein. So, here is a folded protein, that means it forms a specific kind of structure to perform a specific function, but then it can misfold, and then it does not cause that specific or help in the cause of that specific function, and that kind of a misfolded protein often results into various kinds of diseases. Those diseases are mostly linked to neurodegeneration. Neurodegeneration means non-functioning of the neurons. And where are the neurons? You very well know. The neurons are there in your brain, mostly in the brain. And what are their functions? They help in signal transduction, movement of molecules, reception of information, and passing on the information and quickly reacting to information, or even your thinking and all those things. Now, if the proteins are not folded properly, all those cognition functions are gone. So, diseases like Parkinson's, Huntington's, and you might have heard the name Alzheimer's happens because of misfolded proteins. So, these are some of the very interesting facts about proteins. How all these things are dictated? It is dictated by the protein folding mechanism. There are various theories available for protein folding, but not a single one has been singled out or has been accepted or rejected. All of them are there at this point of time. This is going to be my last slide, where I will just give you a glimpse of how people have been trying for years together of deciding how this polypeptide folds. So, let me take you one step back to show you this figure, where as a general biology student you might have heard in your previous lessons that there is a ribosome and there is an mRNA, and on this mRNA the polypeptide gets synthesized. That means, the amino acids gets attached one after the other depending upon the code that is available in the mRNA. So, this mRNA gives code, and according to that code the amino acids gets added one after the other and forms this polypeptide chain. So, once this polypeptide chain is formed, here is the unfolded or nascently synthesized polypeptide chain. So, you have a nascently synthesized polypeptide chain. This polypeptide chain, because it has just come into nature, everywhere it is water and water, some hydrophobic amino acids somewhere here, because they are hydrophobic, they would not like to interact with water. They will quickly try to shy away from water, and as a result what they will do, they will cling on to another hydrophobic amino acid down the lane somewhere. They will say, oh, save myself. So, they will come together because there is water all around. They do not want to interact with water. So, they will come together, therefore, they will form a fold. Then another amino acid somewhere here, they say, oh, come on, I am a charged amino acid, positively charged. I would like to interact with a negatively charged amino acid. Here is a negatively charged amino acid. Let me form a bridge between these two. So, they fold again. So, in this process, it keeps on folding, and then they form some intermediates. After this intermediate folded structure is formed, a final folded structure is assumed by this polypeptide automatically, or by the presence of something called chaperones, which is beyond the scope of this particular subject class. So, it forms a protein which is folded and now functional, and that folded functional protein is called a native protein. And as I told you, at various other instances, this flexible intermediate structure may not always form this protein, although 99% of the time it will form this particular structure, but many times what happens is they may actually form incorrect disulfide bonds or get into incorrect folded structure, and therefore results in an aggregate or incorrectly folded structure. And these incorrectly folded structure results in aggregation of the proteins that leads to various kinds of diseases like the Alzheimer's disease, the Huntington disease, the prion disease, and all those things are listed here. Just the last set of information. So, how is this formed? It is actually derived or defined simply by the thermodynamics. So, what does thermodynamics say? Every structure, every body in general would try to assume a minimum energy structure. So, if you allow me, so why is a droplet a droplet? Because it wants to assume a minimum energy, a spherical droplet, minimum energy structure. So, every body in this universe would like to assume a minimum energy structure. So, here is a funnel, okay, which shows at the top here is the energy. So, energy is very high right here, and then these are some of the tunnels and loops and crevices to which the energy is minimum. So, a polypeptide when it is synthesized has the maximum energy because it has all the loose ends capable of interacting, and because there are large number of loose ends, the energy is very high and it is just flopping around here and there. That energy has to be minimized for the structure to be stable. So, what it does, here it is flipping around, here it is flipping around, it tries its motion down here, down the hill, and ultimately falls into this kind of a structure, which is the folded native structure which is shown here. However, there are possibilities where this structure, for example, which was just flipping around like this, and therefore they constitute this whole energy landscape, finally falls into a crevice which is not the final minimum energy, but somewhere intermediate energy, because it has not gone through this pathway. So, had it followed this pathway, it would have actually resulted into the formation of this native structure with minimum energy, but because this has not gone through that pathway, because it was forming some other kind of structure initially, so it led to a crevice which gets trapped, because to fall into this final structure it has to actually jump over this energy barrier. And now, because it cannot jump over, it has just come and got trapped into a crevice here, which is not the minimum energy structure. This structure is the incorrectly folded structure shown in this set, and these incorrectly folded structure can keep on accumulating, and that will result in the formation of aggregate, resulting in various kinds of diseases. So, that is what I have been trying to impress upon, the structure and function of proteins. With that, I will end this session today and take you to an exciting interesting stories about what are the different kinds of non-covalent bonds that actually help these proteins, or for that matter many other biological interactions to happen. So, at the end I would like to thank the contributors of all the picture and other study material sources that I have taken from different web sources, and here is the list. As you can see, this has really helped me to disseminate this knowledge across the world to many viewers who, and this is primarily intended for educational purposes. Thank you once again for all the contents. Thank you.